Hello everyone, my name is Jillian Heatley, and today I will be discussing with you the forgotten organ. Now you're probably wondering, what organ could possibly be forgotten about? Well, technically, this isn't an organ. It is often considered an organ because of its importance and the role that it plays on our well-being. Uh, this forgotten organ is the gut microbiota. Now, why did I choose to talk about the gut microbiota? Doesn't sound quite interesting, but you will learn today that there is a correlation between the gut microbiota and the food that we eat. Now, I personally have a passion for food and for cooking, and I sometimes wonder maybe I pursued the wrong career choice because I love cooking. And I've always known that what you eat affects your health and affects how you feel. But I never understood the logistics. So in doing this research, it showed me how that happens, and I was quite excited to share it with you guys. So before we get into the nitty-gritty and into the subject at hand, I want to give you guys a little outline of how I'll be going about discussing this topic. So first things first, I will give you a little background of what the microbiota is and why it's important. And then I will go over the physiological impacts that it has. I'll then talk about the methodologies in the papers that I will discuss. And then I will go on to talk about the mechanisms associated with a well-balanced diet, along with the mechanisms associated with diabetes, which you will learn has a correlation with a poor diet. And then I will tie it all together by reiterating the conclusions about the role of food on the microbiome. So, what is the gut microbiota? Well, as I stated earlier, it is considered a complex organ consisting of bacteria, protozoa, viruses, and fungi that actually reside in our gastrointestinal tract. Now, the bacteria in our guts breaks down food that our bodies cannot digest. It also produces nutrients that we need, and it regulates the immune system. Also, it protects against harmful germs. Now, there are many factors that influence the richness and the diversity of the gut microbiome. Now, when I say richness, I mean the abundance of bacteria that reside in the gut microbiome. And then when I say diversity, I mean the different types. The more bacteria, the better. You want as many bacteria, as many different types, and as much as you possibly can get, because the more bacteria, the more nutrients that gets produced, the more protection you have against harmful germs. And as I said, also more help in regulating the immune system. Now, there are different factors, like I was saying, that influence the gut microbiome. And those factors are our environment, medications like antibiotics, and even whether we were delivered by C-section or naturally. And while we cannot control these factors, there is one factor that we can control, and it's our diet. I did not mention it, but it is now emerging as one of the leading influences on the health of our guts. So we can manipulate the balance of our microbes by paying attention to what we eat and being intentional about what we're putting in our bodies. So here is the methods of those papers. So there were two studies that I researched, and they were both done by the same researchers, and they were researching the gut microbiota. The first paper here that I will be talking about was titled "The Relationship Between Hyperlipidemia and the Gut Microbiome of Rats, characterized by high throughput sequencing." Now, if you break down the word hyperlipidemia, you have hyper, which is an increase, lipid, which is fat, and emia is blood, so high fat in the blood. and its relationship to the gut microbiome. Now, in this study, they took a sample of rats, as you can see, the control and the high-fat diet group. And they housed these rats in a specific pathogen-free and temperature-controlled room where they had access to food and water, and they were kept on a 12-hour light-dark cycle. So 12 hours light, 12 hours dark. Now, after these rats were adapted to their environment for about a week, they were randomly assigned to these two groups, either the high-fat or the control diet, or control group. Now, after the 4-week feeding period, 1% of diethyl procarbonate solution was sprayed into the air to eliminate RNA enzymes, and the feces that were passed by all the rats in both groups were collected at the beginning and the end of a 4-week feeding period. And when the rats in the high-fat diet groups showed significant obesity. So after the 4 weeks, at the beginning of the 4 weeks, they collected their feces, and at the end of the 4 weeks, their feces was also collected. And they ran a blood analysis test as well as an alpha diversity test on both of these groups. And I will go further into detail in future slides discussing the results of these analyses, but as you can see, the results are pretty straightforward. The control group had normal levels, while the high-fat diet group had elevated levels. And you will learn that high-fat diets affect the gut microbiota. Various chronic diseases like obesity are thought to be related to intestinal microecological disorders, which can cause major health problems in rats, as well as in humans, such as the development of type 1 diabetes, which then leads me to my next paper, which was titled The Investigation of the Gut Microbiome in Type 1 Diabetes Mellitus Type 1 Diabetes Mellitus rats, based on high throughput sequencing. Now, again, there were two groups, the control group and the type 1 diabetes mellitus groups. Now, in order to create these type 1 diabetes model rats, the researchers injected the rats with the streptozotocin, and they collected the feces. After 1 week of the establishment of type 1 diabetes. So they collected the feces here and ran an alpha diversity analysis, like the last paper. It was quite similar how they went about with the methodologies. And like I said in the last paper, the results are pretty straightforward, although I will explain them further in future slides. But the control group had those normal levels, and the type 1 diabetes had the elevated levels. So, what is type 1 diabetes? Well, it is an autoimmune disease. Its pathogenesis is characterized by the immune system, which produces an immune response against islet. Specifically, it destroys islet beta cells, resulting in the abnormality of insulin synthesis and secretion, and causes the disorder of glucose metabolism in the body. Now, the cause of type 1 diabetes involves a variety of factors, including genes, diet, gut microbiota, and many more. But in addition to the widespread acceptance of the genetic factors in susceptibility of type 1 diabetes mellitus rats, more and more evidence has suggested that the gut microbiome is also involved in the occurrence and the development of type 1 diabetes. Now, here is the alpha diversity analysis that I mentioned earlier. This table is from the paper on the type 1 diabetes mellitus. And when you see these Chow and ACE indices, those are reflections of richness. So like I mentioned earlier, richness is abundance. So the amounts of bacteria in the gut microbiome. And then over here you have the Simpson and the Shannon indices, and those reflect the diversity, so the different types. Now, as I also said, the more, the better. So the more types of bacteria, the better for you, and the more amounts of bacteria, the better as well. So alpha diversity, the results here show that the comparison between the control group here, the first line, and the comparison between the type 1 diabetes mellitus rats, the second line, that there was an increase that was quite evident, and the richness was also increased. So you see here and here, there's an increase, and it's actually consistent with reports that the increase of the gut microbiota diversity is associated with disease. Now here is from that first paper about the hyperlipidemia, high fat in the blood paper, and it showed that you'll see the results here, but after they collected the feces of the rats were fasted for 12 hours and then anesthetized by intraperitoneal injection of 10% chloral hydrate. Now they collected the blood from the abdominal aorta of the rats and then left the samples to clot for 2 hours, centrifuged them for about 10 minutes, and then collected and froze the serum at negative 80 degrees Celsius. Now, using an automated biochemical analyzer, the total cholesterol, which you see here, the triglycerol, these are all fats, the low density lipoprotein cholesterol, and the high density lipoprotein cholesterol were all measured. Their concentrations were measured. And as you can see, the high fat diet group, their levels are all higher than the control group. Just want to emphasize that. And so as we move on, I also wanted to mention the alpha diversity analysis that was done on the paper with the hyperlipidemia rats. So as you can see here, I mentioned earlier, the Simpson and the Shannon indices are the diversity. So here, this is at the beginning of the week, like I said, and this is the end of the 4-week feeding period, whereas the Chow and the ACE were the richness. So there were no differences found in the diversity of the gut microbiome between the two groups before the study. So if you see here, the numbers are quite similar. There's not much of a change or a difference. But after the 4-week high-fat diet feeding period, the diversity and the richness of the gut microbiome were significantly higher than the control group. And that has actually been reported to relate to some type of disease. And as I've mentioned, type 1 diabetes mellitus could be one of those diseases. Now here is a principal component analysis. You're probably wondering what you're looking at, so I'll go ahead and describe. The principal component analysis is a dimensionality reduction method, and it's often used to reduce dimensionality of large data sets. So they do that by transforming a large set of variables into a smaller one that still contains the most information of the large set. So this was done, this was a condensed version of a large analysis that was done, and it's helped us and it's easier for us to read. Now, it was used in this study to describe the differences in the composition of the gut microbiota between the two diets. So the principal coordinate with the highest rate was selected. Now, as you can see, I just want to point out that the red here is the control group at week 0. The green is the high-fat diet group at week 0. The blue is the control group at week 4, and the yellow is the high-fat diet group at week 4. Now, the data shows that there were no significant difference between the two groups and between the high-fat diet group and the control group at week 0. They're quite similar. In fact, they're on top of each other. But while after the 4 weeks of the diet feeding, the flora of the high-fat diet group was significantly different than the control group. So you see the control groups here, but the high-fat diet groups over here. In addition, one thing to note, you think that the control group would have stayed up here, but one thing to note that since the 4 weeks had passed, these rats had aged a whole month, and age actually has an impact on the gut microbiota. Although this was just a month study, if this study were to go on any longer, that's something that you would take note of and actually say that it's quite important. But here, it didn't raise any questions. So, yeah. Now, the conclusions of those studies showed that in the hyperlipidemia paper, in present studies, the diversity and the richness of the gut microbiota and of the rats were significantly increased by the 4 weeks of the high-fat diet feeding. And this difference may play a role in the development of disease. And when we speak of disease, we can then turn to the type 1 diabetes mellitus paper, which stated how the gut microbiome of the rats changed significantly, and even the abundance of the pathogenic bacteria was upregulated. Now, with all of that being said, you're probably wondering, okay, so what does the gut microbiota need? What does it need in order to be fueled? So it is fueled by high fiber diets. Now, fiber is found in most of the healthier foods that you think of. Now, I'll go on to list those foods, but before I list those foods, I'll let you know the mechanism that takes place when eating those foods and how it fuels the gut microbiota. And that is that this fiber gets digested by the bacteria, and short chain fatty acids are produced in the gut that in turn nourish the gut barrier, improve immune function, and can help prevent inflammation, which reduces the risk of cancer. One thing to note, which is actually interesting, is that if you aren't getting enough fiber and you're not fueling this bacteria, the bacteria can turn and start feeding on the lining of your intestines, which causes the inflammation. And that can increase the risk of cancer at the end of the day. So the best fuel for this gut microbiota is foods like fruits, vegetables, nuts, legumes, and whole grains. And the best way to eat these foods are raw or even lightly steamed, but fully cooking them takes out the majority of the nutrients. So you want to make sure that you're getting that pure nutrients that these fruits, vegetables, nuts, legumes, and whole grains have. Also, in a recent microbiome study, scientists found that fruits, vegetables, tea, coffee, red wine, and chocolate were all correlated with increased bacterial diversity. These foods contain something called polyphenols, which are naturally occurring antioxidant compounds, which are very important for the human body to thrive. And so what are the take-home points here? Well, bottom line, diet affects the gut microbiome. And the gut microbiome affects our well-being overall. And I just want to emphasize the importance of eating right, eating the right foods. That is imperative that we eat a well-balanced diet consisting of the proper amounts of nutritional components, such as fiber, that will protect the intestinal barrier. Because one thing to note, you're not only satisfying yourself, you're also satisfying the bacteria in your gut. And you want your gut to be happy so that you can be happy and that you can live a long life. And I just want to say thank you for your time and thank you for watching my presentation.